To start the candy chromatography lab, you first have to know what chromatography is, and it's a process that uses the varying polarities of compounds within a substance to separate that substance, but it can also be used to determine the presence of a substance within a sample. This process consists of two parts: the absorbent, which in this case is the dye sample, and the solvent, which in this case is the eluent solution. The absorbent is the compound that is separated by this process, and the eluent solution is the liquid that carries the absorbent particles up the strip to be separated. This is what a finished chromatogram looks like. The stationary phase at the top of the strip is just paper, and the mobile phase at the bottom of the strip is entirely solvent below the dye sample. In order to quantify affinity with the solvent, you have to find the RF value. The RF value is the distance traveled by the sample divided by the distance traveled by the solvent. The greater affinity that the absorbent has with the solvent, the more similar their polarities. This also means that the absorbent will travel a greater distance along the strip. If the distance traveled by the sample is closer to the distance traveled by the solvent, the calculated RF value will be closer to 1, which indicates that the absorbent has stronger intermolecular forces. So how does this relate to the purpose of the lab? The purpose is to conduct a chromatography investigation to determine the presence of tartrazine in certain colors of M&M dye. Here are two sample chromatograms. In this experiment, the sample containing only tartrazine is the control. You will use the RF value calculated from the tartrazine chromatogram to use as a point of comparison in determining its presence in other M&M dyes. For example, if the RF value of the tartrazine sample is found to be 0.51 and the RF value of the yellow dye spot in the second sample is found to be 0.49, then the second dye sample likely contains tartrazine. To start the lab, you're going to repeat this process with 3 different strips of paper, and try to only handle these strips on the sides and the edges so that the analysis is not contaminated. To begin, you want to measure 15 millimeters from the base of the strip on both sides and then faintly draw a line across. The next step is to measure 9.5 millimeters from the edge of the paper along this line and mark it faintly with a pencil. This is where you will place your dye sample. Repeat this process for 2 more strips of paper. Next, measure 20 millimeters from the top of the strip and fold across. This will create a lip that hangs over the edge of the flask once the paper is placed inside of it. Now take 2 different colored M&Ms and place them in separate wells on the well plate. Now add 1 to 2 drops of distilled water to each well plate. It's important to keep the dye as concentrated as possible, so avoid adding too much water. We also ended up using a brown M&M instead of a green M&M so that it would be easier to see in the video. Now add a drop of tartrazine to its own well plate to use as the control in this experiment, and do not add water to it. Using a clean toothpick, place a dye dot on the pencil marking on one of the chromatogram strips. Keep the spot as small and concentrated as possible when you do this. Do this for 2 different dye colors and the tartrazine sample, so you have 3 chromatograms in total. Allow the dye dots to dry completely before repeating this process 5 to 6 times. Try to keep the dots as small and precise as possible because you want to increase concentration, not surface area, of the dye samples. Use a graduated cylinder to measure 20 milliliters of your chosen eluent solution and pour into a 250 milliliter Erlenmeyer flask. Repeat this process for 2 more flasks before placing your chromatogram strips inside and hanging the lip over the edge of the flask. A watch glass should be placed over the edge of the flask with eluent inside to prevent it from evaporating. Allow the strips to develop for 20 to 30 minutes before carefully removing them and placing them on a towel. Try to avoid dropping the strip into the solvent. Once the strips have been removed from the flask, you want to lightly outline the solvent front, which is how far the solvent has traveled up the strip, lightly with pencil, and outline the colorful dye dot. These are the results of the tartrazine sample and the red M&M dye. Our results are extremely faint, so I'll explain some potential areas for error later on. We're going to find the constant RF value for the tartrazine sample to use as a point of comparison. The distance traveled by the dye was 6.41 centimeters, and the distance traveled by the solvent was 9.49 centimeters, which means that the RF value is 0.675. You will use this value to determine the presence of tartrazine in other M&M colored dyes. Finally, let's get into some do's and don'ts for this lab. Firstly, be sure to place a small concentrated dot of dye on the paper strip to minimize surface area, but avoid adding more than 2 drops of water to the M&M in the well plate to avoid diluting the absorbent. This can make your results extremely faint and less accurate. Next, always keep a watch glass covering the top of your flasks to prevent the eluent from evaporating. Also, don't allow the dye dot on the strip to sit below the surface of the eluent solution in the flask to prevent further dilution of the sample. You must also wait for the dye dot to dry completely before placing it into the flask. Although it's pretty tricky, only handle the paper on the sides of the strip to avoid contaminating the chromatogram or inhibiting the capillary action of the eluent solution. And one last thing to remember when recording data, if your ruler shows up to the 10th place digit, record to the 100th. That's all, and good luck with your lab.